Good morning, students of class 10. So in our previous class we had already started the chapter cell and cell division, in which we studied about mitosis cell division. Do you remember? Mitosis is a type of cell division which is found only in the somatic or the body cells, in which the chromosome number remains same, and it is also called vegetative cell division or somatic cell division. And we also studied that somatic or the mitosis cell division is completed in 4 substages. What are they? Prophase, metaphase, anaphase and telophase. So up to telophase we had studied in the previous class. Now today we have to start with cytokinesis. Karyokinesis we have done. Karyokinesis means division of nucleus. Now we are studying cytokinesis. Cytokinesis means division of cytoplasm. Okay? Cytoplasmic division is called cytokinesis. So I have written in bracket here, division of cytoplasm is called cytokinesis. Now what are the important points about cytokinesis? It results in the formation of 2 daughter cells and it occurs after karyokinesis. So after karyokinesis, that is nuclear division, after karyokinesis, division of cytoplasm occurs. Division of cytoplasm occurs. Means first there will be karyokinesis and then the division of cytoplasm. Okay? And karyokinesis was further completed in 4 substages: prophase, metaphase, anaphase and telophase. Now it results in the formation of 2 daughter cells. It results in the formation of, because of this cytokinesis, 2 daughter cells are formed. In the formation of 2 daughter cells. Alright? And this cytokinesis, it is different in animals and plant cells. So it vary. It vary means differ. The word vary means it differs in plant and animal cells. In plant and animal cells. Means it is of different type. Both of them are not same. The cytokinesis in plant cell is different from that in animal cell. So first we are going to study number A, I am writing here, cytokinesis in animal cell. Cytokinesis in animal cell. Animal cell. Okay, what type of cytokinesis is there? In animal cell, the division of cytoplasm is done by the formation of cleavage. Now what is this cleavage? We will talk about them along with the help of a diagram. Okay? During cytokinesis, the cytoplasm divides by a process called cleavage. The first point you can write. During cytokinesis, in animal cell, the cytoplasm divides by the process called cleavage. C-L-E-A-V-A-G-E. Cleavage. So cleavage formation, how it is done, I will show you with the help of a diagram. What is cleavage? How it is formed? You know that animal cell consists of the outermost covering called cell membrane. And when there is constriction in the cell membrane, let's complete the point now. Second point. During this process, during cleavage, directly I am writing, instead of writing this process, a constriction appears. During cleavage, a constriction appears in the center of the cytoplasm during cleavage or constriction appears. Instead of writing center in the plasma membrane, you can write constriction appears in the plasma membrane of the cell that results in the formation of cleavage furrow. That results in the formation of cleavage furrow. Okay. So second point, I said during cleavage or constriction appears in the plasma membrane of the cell that results in the formation of cleavage furrow. Now the last point we have to do, that is, in this process the cell membrane begins to constrict at the equator. The cell membrane constrict at the equator, resulting in the formation of cleavage furrow. Okay. Now this cleavage furrow, it will deepen gradually. Cleavage furrow will deepen, means it will further go inside gradually and divide the cytoplasm into two parts. And divide the cytoplasm into two parts, resulting in the division of, or resulting in the production of, two daughter cells. Resulting in the formation of two daughter cells. Okay. So what are the points? Now the diagram. So during cytokinesis in animal cells, it occurs by a process called cleavage. Second point, the cell membrane constrict means it will go inside at the equatorial plate, resulting in the formation of cleavage furrow. Here also I mentioned the same. And this cleavage furrow will gradually deepen inside, that it will grow inside, and it will divide the entire cytoplasm into two parts. And once the cytoplasm is divided into two parts, it will be equally shared in both the daughter cells. That results in the formation of two daughter cells. Now look at the diagram which I will draw here of cytokinesis in animal cell. First one. See, this one is the telophase stage. Telophase stage of mitosis cell division. These are the spindle fibers, and here the nuclear material with the centroid and the asters. Okay. Now this one is called cleavage. The constriction which appears, it is called cleavage. Alright, this cleavage will gradually deepen and it will join with each other. Means on either side this cleavage will come, it will go deep inside in the second stage, and it will join to form. Now see, the constriction appears here. understood? Now this one is two daughter cells. These are the nucleus. This is the cell cytoplasm. The cytoplasm is completely divided into two parts due to the formation of this cleavage furrow, which has almost joined each other. Okay, they will join each other and cut the cytoplasm into two equal halves. So it results in the formation of two daughter cells. These two are called the daughter cells. Alright, so from the one parent cell, the cytoplasm divides equally to form two daughter cells with equal amount of cytoplasm in both of them. And this process is called cleavage furrow formation or cleavage. Okay, so process of cytokinesis in animal cell is completed by the formation of cleavage. Got it? Okay, so now next one is cytokinesis in plant cell. Now number B, cytokinesis in plant cell. Now how cytokinesis is done in plant cells? Okay, in case of higher plants, there is a special mechanism for the formation of or for the process of cytokinesis. So we'll write it in points first, and then along with the diagram you can understand how it happens. It occurs by a special mechanism in higher plants by a special mechanism in higher plants in which the cytoplasm divides or is divided into two parts by the formation of cell wall in the middle of the cell. Okay, so what is the first point I wrote? Cytokinesis in plant cell occurs by special mechanism in case of higher plants in which the cytoplasm is divided into two parts by the formation of a new cell wall in the middle of the cell. That is known as cell plate. So it results in the construction of a new cell wall, that is called construction of new cell wall. I have not written, and that new cell wall is known as cell plate. The structure which results in the division of cytoplasm in plant cell is called cell plate. What is cell plate? It is a new cell wall, and from where it is produced? It is produced from the plants itself. Okay, so now the next point that we have to do is the diagram and then the remaining points. Now look at this plant cell. This is the cell wall, then the cell membrane inside. Now what I will draw is, I will draw the two nucleus here because only cytoplasmic division is taking place. Now draw small microtubules. They are the microtubules with the spindles here. Okay, so what is this? This one is the nucleus. Now what is this one? Outermost layer. It is called cell wall. Let's label it. This one is the cell membrane, of course, the second one. Cell membrane. This one is the nucleus. This one is the cytoplasm. And this one, it is known as membrane bound vesicles. Membrane bound vesicles. Alright, membrane bound vesicles. Now next diagram you can see. the same plant cell with the cell wall and the cell membrane. You should not touch it, okay? And now in the middle we have the formation of new cell plate. This is a new cell wall with the nucleus on either side. Finally, in the last stage, we find like this. You can draw. This one is one cell. This is the with the nucleus, and this is another cell with another nucleus. Okay, two cell wall. The partition is fully formed now. Understood now? Got it? We have two different daughter cells. These two are called the daughter cells. The newly produced cells are known as daughter cells. This is a cell plate. So if you want to mark it, this one you can write it as cell plate. Got it? Just have a look at the diagram. Cell wall, cytoplasm, nucleus, membrane bound vesicles, this one. Okay? And cell membrane, already I have said. This one, this structure is called the cell plate, and the newly formed cells with the equal amount of cytoplasm in both of them, it is known as daughter cells, because from one parent cell two daughter cells are formed. Now next point, only one point was completed here. Okay, next point is the polar spindle microtubules. How the cell plate is formed. The polar spindle microtubules. What is this microtubules? The one which results in the formation of spindle fibers. So polar means you know that polar region, this one like you cell is there. This is called the polar region, the upper part and the lower part. No, not pole, south pole like that. The polar spindle microtubules assemble in the middle plane. Assemble means they will come together. Assemble in the middle plane. Here, they are coming in the middle plane. All the polar. This one, this microtubules assemble themselves in the middle plane and forms a cylinder-like structure called phragmoplast. So I have not marked here phragmoplast. This structure is called phragmoplast. Phragmoplast. The polar spindle microtubules assemble in the middle plane and forms a structure called phragmoplast. This is the precursor. Phragmoplast is the one which results in the formation of cell plate. So next point that you will write is small membrane bound vesicles. This one is the membrane bound vesicles. They are derived from Golgi apparatus. So I will write it. And filled with, what they are filled with? They are filled with the cell wall precursors. They associate with the microtubules and they form the early cell plate. So next point you can write it. First we'll draw it. This diagram already complete, so no need to keep it now. The small microtubules. The diagram was already completed, so that's why I rubbed. A small microtubules, a small membrane bound vesicles I have to say. Then microtubules later. The small membrane bound vesicles bound. The small membrane bound vesicles. Bound. The small membrane bound vesicles derived, that point I have not said Golgi apparatus, that time I said no. That small membrane bound vesicles derived means they are obtained from Golgi apparatus. Golgi apparatus, what is that? It is the name of a cell organelle. Okay. the small membrane-bound vesicles derived from Golgi apparatus and filled with cell wall precursors. Cell wall precursors means the one which results in the formation of cell plate or cell wall or cell plate precursors, cell wall precursors. They associate, means they will combine with the microtubules, associate with microtubules to form the early cell plate. Okay, so small membrane-bound vesicles, as it is, I had already shown you in the diagram also. This membrane-bound vesicles like this and like this here and drawn. No, so they are the membrane-bound vesicles. From where they are obtained? They are obtained from Golgi apparatus. What is Golgi apparatus? You know, is the name of the cell organelle, and it is filled with these membrane-bound vesicles. Means they contain the cell wall precursors. Means they will only result in the formation of that cell plate. And they associate with the microtubules. Microtubules are there in the spindle fibers, which helps to form the spindle fibers, and they result in the formation of early cell plate. Means the previous or the ancestrals, we can say the starting of the cell plate is formed because of them. Understood? So once the cell plate is formed, now the partition of cytoplasm is possible, and that will result in cytokinesis of plant cells. So now the next topic we have to study is significance of mitosis. Why mitosis is important? At least 4 or 5 points you need to learn about the significance of mitosis. Significance means importance. So why mitosis cell division is important, or what are the benefits? So next heading is significance of mitosis. Okay, first point. Mitosis cell division, it results in the formation of two daughter cells with same number of chromosomes. So we can write, it helps to maintain the chromosome number in individuals. It maintains the chromosome number in the individuals. Okay, like if our body cells has 46 chromosomes, then after undergoing mitosis cell division, the newly produced body cells will also contain the same number of chromosomes. Number 2, it helps in growth and development in different parts of plants and animals. Help in growth and development in plants and animals, because it is mitosis cell division which will help or which will produce the new body cells, and once the body cells increase in number, then only the growth will be possible, either it is plant or animal. And the third point is, many unicellular organisms like amoeba, bacteria, they can reproduce asexually by the method of mitosis cell division only. Directly the cell will divide, and that will result in the formation of two daughter cells, like binary fission in amoeba. So third point you can write is asexual and vegetative reproduction in eukaryotic organisms. Asexual or vegetative reproduction in eukaryotic organisms. Now the fourth point, or the fourth significance of mitosis cell division, it helps to replace damaged cells of the body by producing new cells, by regenerating the new cells. So next one is it helps to heal the wounds at the wound site. It helps to heal. Heal means to fill. Like if you have any wound, any cut is there in the skin, after a few days the skin will join due to the process of mitosis cell division, because the cells will divide, they will grow, and they will try to fill that gap or the wound. It helps to heal the wounds at the wound site or injured site, place where wound is there. Then the last point, number 5, it maintains the chromosome number in the body. Already I have said, okay, it helps to repair the damaged cells of the body. Means repairing is also done, regeneration is also done. So that also we can write, it helps to repair and regenerate the lost parts of the body. Okay. So now next we have to do is differences in mitosis. So we have understood the significance of mitosis. Once again, it maintains chromosome number in the individual. Like if the parent cell has 46 chromosomes in our body cells, the daughter cells, the newly produced body cells will also contain same 46 number of chromosomes. Number 2, it helps in the growth and development in plants and animals. Plants are able to grow in height. It's because of mitosis cell division. Third one is asexual or vegetative reproduction in eukaryotic organisms. You know what is eukaryotic? All the modern organisms like plants and animals are eukaryotic, which has a well-defined nucleus with a nuclear membrane. So they can reproduce asexually by this method of mitosis cell division. Example, binary fission in amoeba, in which a single amoeba cell or the parent cell can divide to form two daughter amoeba. It also helps to heal the wound. Heal the wound means if there is any injury due to accident, then those broken or damaged parts of the body at the wound site can be healed by the process of mitosis cell division. And the last one, it helps to replace or repair the damaged and broken cells of the body. Okay, so whatever broken parts are there, once again they can be regenerated. Okay, now next is the differences between mitosis in animals and plant cells. So I will write here differences between mitosis in animal and plant cell. What are the differences between these two? Okay, so we can draw a table like this. This side we have the animal cell. On the right side we have the plant cell. Now, first difference between animal and plant cell mitosis. Number 1, in animal cell, what are the differences between mitosis in animal and plant cell? Under animal cell we can write, during mitosis, the cytokinesis occurs by cleavage formation. So first point, you can write centroid is present. In plant cells, there is no centroid, no centrosome, because centrosome is the cell organelle which is found only in animal cell, not in plant cell. So centroid is present and help in spindle formation. Centroid is present and helping spindle formation. The spindle formation is required during cell division. Alright. And here in the first point you can write, there is no centroid. Then how the spindle fibers are formed? Spindle fibers are directly formed from the microtubules. So centroids are absent and spindle fibers are formed by the assembly of microtubules directly. Centroid is absent spindle fibers are formed. By the assembly of microtubules directly. Okay, centroil is absent. Spindle fibers are formed by the assembly of microtubules directly. Means it is the microtubules which result in the formation of this spindle fibers. There is no centroil. In plant cell there is no centroil because centrosome is absent, so centroids are also missing. Second difference you can write, asters are formed because centroil is there, like this is a centroil, and radiating rays are also produced, they are called the asters. So here asters are also formed, so we can write asters are formed. Second point, asters are formed, and here in the plant cell asters are not there, so asters are not formed. That's why this type of, like the mitosis division in plant cell, is also called an astral division. So asters are formed here, asters are absent. Asters are absent, so mitosis in plant is also called an astral division. Okay, an astral division it is called. In case of plant cell, the type of division is called an astral. And the third point you can say about cytokinesis. In animal cells, cytokinesis occurs by cleavage formation. So third point is cytokinesis occurs by furrow formation or by cleavage formation. Cytokinesis by cleavage or furrow also you can write formation. And here you write cytokinesis occurs by cell plate formation. Cytokinesis occurs by cell plate formation. Okay. So what we studied today, now this table, it is the differences between animal cell and plant cell in terms of mitosis. Three differences are there. What are they? First one is animal cell contains centroil that help in the formation of spindle fibers. Plant cells, it is absent, and the spindle fibers are directly formed by the microtubules, which assemble means they come together directly on the polar region. Then second one is asters are formed in animal cell around the centroil. The asters are there, radiating rays just like the sunlight is there. In case of plant cells, asters are absent, and so the type of mitotic cell division in plant cell is also called an astral division. And the third one is cytokinesis occurs by cleavage or furrow. In plant cell, cytokinesis occurs by cell plate formation. So what we studied in today's class, you can take a recap. Like we started with cytokinesis. What is cytokinesis? Division of cytoplasm. Then we studied the steps of cytokinesis in animal cell and plant cell, and finally the differences between plant cell and animal cell on the basis of cytokinesis. So in the next class we can continue with the next type of cell division, that is meiosis cell division. Thank you.